it's my great pleasure to introduce Christina Kudalbo, who will be giving a short introduction on the two speakers. And I'm very happy to have you here, and let's have a great day.
Hi, I'm Christina, and I'm very happy to welcome you to our 34th CIBM Breakfast in Science. And today we have speakers. So I will start by presenting and introducing the first speaker. The speaker will be having 20 minutes to present, questions, and I will be introducing the second speaker, and so on. And then at the end, if the time, if you need more time, we'll have to for questions. So the first speaker is a dear colleague and collaborator with whom we worked closely in the past months, near Bernard. So her name is Paola Ballesteros, and she's a doctor. She has a PhD in neuroscience, obtained at the National University of Mexico in Mexico City, Mexico. And she's working very closely with Professor Marie-Catherine Lausanne from CHUV and from the laboratory of Radioncology. So her lab in Mexico was the first lab who was looking at the effects of radiotherapy on the brain, long-term effects after having a brain tumor and after having undergone radiotherapy. So today she will be talking about targeting brain tumors in Milliseconds, the potential flash radiotherapy to minimize side effects. So Paola, thank you. Yours.
So yes, I'm going to talk to you about the flash radiotherapy, which is something very new, or recently new, and its possibility to treat the brain tumors without that much side effects as conventional radiotherapy. So we know that radiotherapy is one of the most important treatment cancers we have. At least 50% of all the cancer patients will receive radiotherapy at some point. And of course, the idea is to kill the tumor cells with radiation, ionizing radiation, but we are limited because of the adverse events that can happen. So at certain points, of course, luckily the tumors can respond and are like less resistant, but we also are limited always because of the normal tissue response. So the conventional radiotherapy is now established in very conventional schemes that are known clinically. Normally there are several fractions, normally it's 2 grays of dose per fraction and per session, 1 session per day, 5 days a week. And the dose rate, conventionally, it's 2 gray per minute or 0.03 gray per second. And the size of the field is centimeters. So how the physical dose, the energy that is delivered in the tissue translates to the biological response depends on many parameters of the dose, like the type of irradiation, the beam energy, the spatial distribution of this irradiation in the tissue, the temporal distribution, and the dose rate, meaning how fast this energy is delivered in the tissue. So we knew for a long time ago that the dose rate was important because here is the clinical dose rate we normally use, but if we go so slow, we may compete with repair of the damage in the cells. So we give some time to the cells to repair. So we know this already. But what happens if we go very, very far away, like 3 orders of magnitude higher in the dose rate? What could happen? Well, this is the flash effect. It's like giving the total energy in a flash in milliseconds to the tumor very, very fast. So flash radiotherapy is irradiation at ultra high dose rates, very, very high. And the shift goes from minutes, the conventional treatment, to milli or even microseconds. And of course, this also improves the movement management because it's so fast that the patients don't even have time to move. So this is also an advantage of this kind of radiotherapy. And what happens when we go that far? Well, what happens if we protect again? So we saw that if we go very low, we protect because we give time for repair. But if we go so high, what we see is the survival here, for example, at a certain dose, the survival of the cells is higher when we go very, very fast in the delivery of the radiation. So what actually is happening is increasing the therapeutic window we have to treat the patients, the difference between the response of the normal tissue and the tumors. And luckily, the anti-tumor effect, its efficacy is preserved after the flash. So this is called the flash effect when in very ultra high dose rate, we maintain the anti-tumor efficacy, but we reduce the normal tissue complications. So this is very, very intriguing. And it's not really new. It has been discovered in the 70s. This is very old experiments in the skin, when you can see the side effects in the skin, the scale. And you see here, like for a conventional dose rate, we have like a very bad reaction, but if we go very fast in the dose rate, the reaction is reduced. So this was already being described, but what they thought is that maybe we will protect also the tumors. So this field is stand by like for 4 decades, and no more research was done because they thought, okay, we are protecting cells, but we would protect tumors, and you don't want that. Until the 2007, the group of Marie-Gat Bossanin in Paris with Favodon, they start doing research again with this very ultra high dose rates with the experimental machine, and they were trying to study fibrosis in the lung. And what they found is that there was no fibrosis in the lung after very high dose rates. So they replicated a lot of experiments until they decided to publish in 2014, and this was like rebirth of this flash field. And here you can see the pictures how the normal tissue of the lung looks exactly the same as the flash irradiated, but the conventional shows fibrosis at the same dose. So this is very, very intriguing. So from this date to our days, a lot of people have been able to replicate these results in different type of models, not only mouse, but only pigs, cats, and zebrafish. And they have shown protection in different type of tissues. And we have also seen that there are some reports that don't found this effect. So this means that there are some parameters in the beam and in the dose rate and the structure of the beam that may be influencing the presence or not presence of this effect. So this is still ongoing research. And luckily, we don't protect the tumor. So we have seen that we can protect lung, gut, skin, and brain with ultra high dose rate, but the tumors, we are not protecting them. And for different type of irradiations, electrons, protons, X-rays, and C ions have proved the same effect. So why this is happening? Well, we know that radiation interacts directly with the tissue, producing free radicals in the water of the tissue. And these free radicals can damage all the structures, but also can have a direct ionization in the proteins of the structure. And if we damage the cell, this will finish in cell death. But there's a big step between the first steps, the chemical step, the biological step, and the toxicity we can see in the long term. So we are still investigating to understand what is happening with this flash. What we know so far is apparently in some models we have a less DNA damage. For example, at least in this intestinal crypt results, we can see that the foci of damage in DNA are reduced in flash compared with COMP. We also have seen there's a reduced cell death. This is the same experiment in the lung. And you can see here, this is a marker of cell death. And you can see how it's reduced in flash compared with conventional. Apparently also the cell proliferation is preserved after flash. This is a proliferation of new neurons. This is double porting, a marker of immature neurons. And this is a BRDU, marker of proliferation. And again, you see that controls and flash look pretty much the same. And also the inflammatory response induced by irradiation is reduced. This is a marker of microglia and activation of microglia, and the colocalization of the microglia and the activation, it's again reduced in flash compared to conventional. And also the vasculature seems to be well preserved after flash. This is a colocalization of lectin, which can show the vessels, and again the inflammation. And you see again the same trend that for conventional, the damage is reduced in the flash. We also have some evidence that lipid peroxidation, the peroxidation of the membranes and all the lipids is also reduced in flash. And also the mitochondrial damage, meaning the damage, for example, this is in fibroblast, long fibroblasts irradiated with protons, we see again a reduced damage in the mitochondrial matter. So we are protecting the tissues. We still don't know exactly why, but as we can see in this diagram, We have this reduced DNA damage apparently. We are preserving the stem cells and progenitors with FLASH. We have less vascular damage. We have less inflammatory response, and we have a better preservation of organ function. So the big question was, well, it's going to protect the tumors then, but it seems like it's not protecting the tumors. It's not better than conventional, but at least it's the same. It's iso-efficacy of the FLASH compared to conventional in the tumor response. This has been shown for several kinds of tumors and also different types of irradiations. So what we wanted to know if this FLASH effect could be present for medulloblastoma tumors. This is because medulloblastomas are the most common malignant brain tumors in children. And this is a tumor that grows in the cerebellum, and the incidence is in very small children between 3 and 9 years of age. So what we did is try to study this. So this is the standard treatment they receive. They normally receive surgery, then radiotherapy and chemotherapy, and they give a dose between 18 and 24 gray, and sometimes additional boost to the tumor bed. So this is very good for kids. The survival is really high, 72%. But there's a lot of problems in these patients related to the cognition, the endocrinology effects, and neurologic effects. One of the main effects is the cognitive decline. So years after radiotherapy, these patients can have problems in cognition, memory, processing, attention, and executive function. 90% of the survivals may require accommodation at special schools for special learning disorders. And for example, you can see that there's a decline in the IQ quotient of 20 or 30 points after certain dose. So this is a very high problem that exists in patients. So of course, we wanted to know if the FLASH effect was possible for these kind of tumors, because it's important if we can reduce the cognition damage. So for this, we use a cell line called UW228 that comes from a 9-year-old female girl, and we implanted these cells in the cerebellum of mute mice to make them grow. So after establishing this model in pediatric mice, we irradiate them and we do the follow-up. For the irradiation, we're using this machine, the CHUV, and we're using a 3x stem fractionation scheme, both in conventional and FLASH dose rates. You can see here the difference in the dose rate is very, very high. So we do the cells injections in the cerebellum with 1 million cells in total microlittle total volume, and we do the follow-up using CT with contrast to see how they grow up in the cerebellum. And we also do some histological analysis to see how these cells are behaving. So what we have seen first is that there was no difference between males and females in the tumor growth with the wicks in the cerebellum. And we also found that both conventional and FLASH radiotherapy can control the tumor growth completely. So with this after 6 months, they are still, they were cured completely. So this means that both FLASH and conv are iso-effective for the control of the medulloblastoma, which is very good news. And you can see how for all the non-irradiated ones, all the tumors grow at some point. So something important is that the survival was again very well and non-statistical significant difference between conv and FLASH irradiated in the survival, which is not the case of the non-irradiated ones here. Another big question is about the long-term effects. So are we reducing the long-term effects with the FLASH effect? So for this, we did behavioral analysis, and another group with California, which collaborates with us, has done also electrophysiology and immunofluorescence in these mice. So for behavior, what we do is called novel object recognition, which basically puts the mice in an arena for habituation. And then we put 2 objects, and the mouse will explore the object, and we count the time that mouse is exploring each object. If we change one object, then the mice will recognize which one is the new and explore more time the new one, if it's remembering. If it's not remembering, then we have a different in the discrimination index. So here you can see how the FLASH and the controls again look pretty much the same, and it's not the case for the convention. And the electrophysiology, we use the LTP, which is surrogate marker of memory. And you can see if we stimulate the neurons of the hippocampus, normally what we expect is establish the memory, like here, like in the green, dark green. And it's not the case in the COM, where the memory is kind of reducing in time, the response, the electrophysiological response. So again, flash is protecting the neurons. Also, if we can check the integrity of the synapses with these two markers of synapses, which is pre and post-synaptic markers, we can see again the same trend, that control and flash look pretty much the same. And this is a protein of synaptic vesicles, and again, is well preserved with the flash. So we want to know if we can also see this protection of the brain in MRI imaging after this flash. So what we did is use irradiated mice with one dose of 10 gray, and then 10 months after irradiation, when the cognition effects are more relevant, we did the in vivo brain imaging using the 14T, and then we sacrificed right after the imaging, and then we performed the same mice the ex vivo brain image. So first we do a pilot with the 4 controls, 3 comp and 3 flash. And then we repeated this after setting all the parameters with the group of Christina and all her team. And we did six controls, 6 in the conventional irradiation and 6 on the flash. And these were 30 months after irradiation. And then we repeated the ex vivo imaging. We controlled the time of the PBS the same for all the mice. So for this, we use the 14T you have here that you might know very well. And we use all the techniques that have been established by Christina's team, using the fixation of the head and the monitoring of the breathing and the temperature during the whole anesthesia time of the mice for the in vivo. And we did some structural sequences and also DTI imaging. And for the ex vivo, as I said, we perfuse the brains, and then we use the same PBS regularization time for all the mice. For the ex vivo, we included the 2 brains each time in fumbling using this device designed by Johan, and we extracted all the bubbles, and then we put them again in the coil to do the imaging. The difference between the in vivo and ex vivo, well, it's almost the same. We're having 2 structural sequences and the DTIs, but since we don't have the limitation of the anesthesia time for the ex vivo, of course, we did more directions and a higher intensity of the V values during the whole night. So what about it has been reported in the hippocampus MRIs after whole brain radiotherapy? It's known that, for example, the hippocampal volume in irradiated, it's 5.7% reduction 7 months after irradiation. So this means that we look for some structural reduction of the hippocampal, very basic. So we know also another cognition impairment models like Alzheimer's disease have shown a reduction in the volume of the hippocampus. So very basic volumetry we did in the hippocampal volume. And we see a little reduction in the COMP, and what was not significant in the flash, but there was no significant difference actually between both. But you can see that there's a reduction in both cases of the hippocampal volume after the irradiation, a little bit higher in the COMP, but it was not statistically significant. But about the DTI, we know that this DTI is a non-invasive way of understanding how the structural connectivity is preserved after irradiation. Of course, it gives us an idea of how the axonal organization macroscopically it's doing. Of course, we are following the water molecules and the diffusion in the brain, and this will give us an information of how the fibers may behave, but also the neurons. So we know that water basically moves a Brownian movement, but if it's restricted by, for example, the axons, it will have some specific directions. So it will change from isotropic diffusion to everywhere to a specific direction, anisotropic diffusion. And we can measure this with the DTI, basically. So the basic indexes of diffusion, for example, is a fraction of anisotropy, which can be 0 when the diffusion is spherical, or one when it's tubular, for example. And another traditional imaging diffusion tensor metrics would be, for example, the mean diffusion, the radial or the axial diffusion, or the ADC, which can also give us different information of how the water is diffusing in the brain. So this is basically different metrics we can use. But recently, in collaboration with Craig Stark in California, he's using another model to analyze the diffusion. It's called NODI, which is Neural Orientation, Dispersion, and Density Imaging, which basically, instead of using these ovals for the calculation of the direction, we're using more complex structures. And this gives us like more detail in the small pathways, to say. So like here we can have like a bigger pathways, and here we have a very, very small diffusion pathways in many directions. So it uses three compartments. One is for the cephalorectilian liquid and the neural density index. And this would correlate, although it's not very directly, but with the dendrite density. So it's related to the connectivity of the brain, but in the gray matter, in contrast with the DTI conventional, which is more related to the white matter. So what about the DTI in patients after radiotherapy? Well, it's known that the DTI can actually predict the cognitive decline after radiotherapy. This study, for example, has shown that if you do the MRI before and after radiotherapy, the results in the DTI can correlate with the cognition in the long term in these patients. So in this study, for example, they have seen that in the parahippocampal cingulum, in this very small structure connecting the hippocampus with the cortex, if you have differences in the DTI, this can predict the decline, as you see here, in the verbal fluency 18 months after radiotherapy. So this means that we can see these changes in the diffusion or the connectivity very early after radiotherapy, and they could correlate with the cognitive decline. So we wanted to see the same, of course, in the mice, if we can see the same in some of the structures related to the hippocampus. So I will show you this results we have. These are kind of preliminary results obtained by Craig in California. We have results of the in vivo, and he used 4, 5, and 5 because the other ones were a little bit problematic. The images in vivo, and here you can see the apparent diffusion coefficient, the ADC, and these were co-registered all together and do the registration and alignment. And then he did basically several of t-tests between control and conventional. So you can see here, if it's 1, that means that the control is higher. And if it's cool, if it's blue, the control is less. So here you can see this very big picture of what is happening. So what you can see here is, for example, control and conventional. Here you can see there are some areas surrounding the hippocampus. For example, this is the fimbria, where you can see a difference between the control and conventional that is not really present between the control and the flash. And if you do the control and conventional, it's a smaller difference. So this, you can see that at least the big picture is that maybe something there, but still we need to analyze each region separately. So this is ongoing analysis. To select, for example, we have select now the hippocampus, the whole hippocampus, and some other structures related, for example, the fibers and smaller volumes to do the analysis with more detail. So this is, I will put here that this big difference we don't trust much because it's a little bit too low. So, but we can see some difference there, but not really much. So again, with the free water diffusion, which is related to this liquid, we can see again some differences in some regions, again, between control and flash. We don't see much, but we can see between control and conventional. And also here, for example, near the hippocampus or near the fibria, but also here in the caudal nucleus, maybe. So there's something going on. And about the OD analysis, which is more related to this dendrite of the gray matter, you see how the putamen may seem a big difference, for example, between flash and conventional. So we have something there going on in the diffusion. but we still need to do more analysis of this. So this is very preliminary data. And about the ex vivo results, for us it would be very nice if the conclusion would be similar at the end of all this study in the ex vivo compared with the in vivo, because of course ex vivo we don't need all these ethics and it's easier somehow to do. So it would be nice to have these markers in the ex vivo brains, which would be more easier to do. So basically what we have seen, of course, we cannot have the water, but we have the only dispersion index. And we have some difference here between flash and conv, more in the cortex and also in the hippocampus area. And the fraction that anisotropy, we have also some areas that you see that the difference are reduced between the controls and the flash. And you see more areas between the flash and the conv and the control and the conv. So this is our preliminary data, but I will say that we need to analyze more in collaboration with the group of California. But what we have seen so far is that the flash effect, meaning the iso-effective response of the medulloblastoma tumors is possible. And also there are some indications that we are protecting the cognition also. So this means that this flash effect, it might be possible for the treatment of the medulloblastoma tumors, according to our results. And what we still need to define how to do more into the clinics, how to go into the clinics. And we are planning to do these MRIs because these MRIs were performed in adults for this first study. So we will in the future perform the same study in the Swiss Neurology. We have already started this batch and we will continue with this work to see if we can see some difference in the pediatric mice. So this would be all, and thank you so much. I want to thank, of course, of the Christina Kudaldu and team, because they have been very helpful, and of course they have also been very interested in this project, and of course the team of Marie-Catherine Bosnan and the physics team, and also Charlie Limoli team and the Fred Stark team. Thank you so much.
Are there questions from the audience? And then, yes.
Very, very interesting. Maybe it's a naive question, but at some point you showed a lot of studies using electrons, protons, etc. But for this flash technique, what works better? What is more suitable? Because as far as I remember, the position of the dose is completely different. Yes. Either you use protons or electrons. So what is better?
Well, so now we always use these machines that are still experimental, so they are not very conformal, meaning that in the clinics you can shape the shape of the beam actually to conform the tumor. And these machines are still all prototypes, and even for protons they are modifying some machines in the clinics, but this is kind of more clinical significant. So the response in the tissue, it seems to be the same. So until now, we don't see a very clear difference, but this is, of course, ongoing work. So we don't know if the protons will be better than the electrons in terms of the flash effect. But we know, for example, that protons are more conformal. So protons can protect more the surrounding tissue, even in the conventional. And also, it's true that protons, it's kind of easier to modify the dose rate in the clinics, for example. So maybe it's more easy to go in protons to the clinics because electrons, they cannot go very deep. So this is one limitation. So they cannot treat very deep tumors. So for example, if
MB is a deep tumor, or
sorry?
The MB tumor is a deep one.
The medulloblastoma, you mean? Well, this is, well, for mice, of course, it's not deep. So for us, it's good for the electrons. But yes, for the patients, they may need protons or photons, which is also in progress. So this photon flash therapy is also something that I think we would have seen at some point.
Yeah. Other questions? Because I have some. So if I understood correctly, so now on the mice, you irradiate the entire brain more or less.
Yes, we
And you are looking at different brain regions. And the idea is to see now with the flash therapy that the brain is somehow protected
Yes,
compared with the conventional radiotherapy on healthy mice.
exactly.
The tumor that you showed us is a tumor who develops mainly in the cerebellum.
Yes,
So I guess at one point we'll be bringing mice with the tumor in the cerebellum.
yes, I hope so.
Yeah, so the idea would be, so me, I'm starting to think now further on, is that we will have to look a little bit at how the tumor looks like, because we would like to see, based on the presentation, that the flash therapy will kill the tumor, like the conventional therapy, but will protect the other parts of the brain, something that the conventional therapy will not do so well.
Exactly.
Okay, so that would be more or less, so we will be looking, so you will be again irradiating the entire brain,
Yes,
with the hope that we will be seeing the decrease of the brain tumor and completely elimination of the brain tumor based on the results that you showed,
yes.
and that the other part of the brain, so we will,
It's more
so we will be doing spectroscopy in the cerebellum, but also in another brain region out, like the hippocampus or cerebral cortex.
It will be challenging.
Yeah, and the diffusion and brain volumetry of the entire brain.
Yes, exactly. This is important because in mice we can take out the brain and see the flash effect, or we can do some cognitive very long term. But if we have something earlier time points, for example, in the MRI that we can see that flash is protecting, then it would be easier to see in patients in the future, because this would be nice. Not to have to wait like 1 year to see the cognition is preserved or not, for example. Because cognition is the problem, that it's very long term and it's also very tricky.
Yeah, and also the mice, they become older with the time.
Yes, exactly.
Humans.
Yes.
So cognition
Exactly.
will probably accelerate with the age of the mouse.
Exactly.
So then another question, because I saw.
It's Olivia.
It's Olivia, yeah, who's asking the question.
Okay.
Yeah, hi, Olivia.
Hello. Thank you for this really interesting talk. I was wondering about the dose rate. Have you identified an optimal dose rate? Because I saw that you use several dosage. And also, is this dose will be animal or patient dependent? And on what parameters will it be optimized?
Yes, I will start with the first one about the animal and patients. We still don't know. Only one patient, a human patient, has been treated at the CHUV, and it was for a skin lymphoma. But this is a squamous skin lymphoma, and this is the only data we have. But we have some ongoing trials now running at the US at Cincinnati hospital. So we will see how it goes with humans. But yes, this is a big question if we can see the same. And about this, let me see if I can share some image I think I have about the dose rate, because this is also a very important question. So let me share this. So this is some data that has been published in a paper. So here you can see how the irradiation, total time of the irradiation, of course, is very fast in the flash, but also the dose rate in the pulse is changing. So you have different combinations to do the flash. So you can see here that apparently there's some kind of range. Here you see the flash effect, and here you don't see the flash effect in this X. So of course there's apparently a limit here that when you have the flash effect and you don't have flash effect. And the total durations of this would be around milliseconds, the limit. So if you go higher of tenth of milliseconds, then you will start losing apparently the flash effect. So yes, there's some limit in this, but this is a very, there are a lot of people in the world trying to find exact parameters to define the flash effect. And I think we will have a very new results soon, about this.
Thank you.
My not very last question, but maybe afterwards I can come with the other ones. You showed that Professor Limoli is using in University of California he's using this, he's also irradiating the mice, and he's doing more behavioral tests or more cognitive tests.
Yes.
Is he using the same machine?
Yes.
How difficult is to get the same machine, because it's really
Well,
yeah, it's not
he's using the same machine in here. So, no, so he's using exactly the same machine that we are doing. So we are irradiating the mice here and sending them by plane. So they have an extra stress, of course.
Okay.
And he's doing the analyze there.
Okay, so he's
Yes.
okay. So the mice are irradiating here.
Yes.
And so there. Okay, that's a challenging.
Yes,
that's a
definitely.
really intercontinental project
Yes,
with everything that involves mice translocating.
Exactly. The beta. Okay, controls extra stress.
Okay. Manar?
Yeah, maybe I have an opportunity to discuss it maybe a bit deeper, but I wanted to know if you could say a few words on how you would explain the difference you had on the batch effects and batch tissue compared to tumor. How is it linked to physiological parameters?
Yes. This is the big question that we see in all the meetings about the subject. Let me see if I can find the. I think I have some. Well, one of the main hypotheses for a long time was the oxygen, because we have seen, for example, that I will show you this one, that when you increase the oxygen, you put carbon breathing to increase the oxygen in this mice, the flash effect is reduced. So this is something related to oxygen. So one of the first hypotheses was that oxygen was being depleted, and it's known that radiotherapy with low oxygen, it's damaging less. But this has been shown that there's not much depletion actually in the flash compared to the pump. So basically, what I think it might be related to also the perfusion of oxygen. But there's many hypotheses there. Some of them are related to the recombination of radicals, that is, since it's so fast that the recombination rate is different in the free radicals that it's occurring. Another hypothesis says that it has to do with the circulating lymphocytes. So since you have less, the conventional is taking longer time, so you are irradiating more lymphocytes than the flash, so then that's why you are protecting. These are many hypotheses. Some of them are more accepted than others, but still ongoing work. My bet is that it has to do a lot with the reperfusion of the heart, meaning that you don't have much time in the flash like to reoxygenate the tissue. But of course, this has not much influence so far, but we need still to do some testing. So this is something that we are ongoing. We're doing some modeling, and also we will do some experiments to test and to see what is happening. And this is another about immunological response. So this is another hypothesis about the lymphocytes. But at least in our group, we don't see a lot of difference in different status mice, for example, nude mice compared to black. So probably the lymphocyte hypothesis may not be the best one. So this is what we think. But yes, there's many hypotheses. Yeah.
I have some ideas to do. I also discussed with Sean about it. So what about doing some direct oxidative stress measurement issues?
Yes.
So we have a machine here, the DTR spectrometer,
Yes,
the bench top, and will allow to really measure different brain regions.
Yes.
And to see how is the oxidative stress, and you can look at different parts
Yes.
in the oxidative chain to see which are effective and which are not. That will be interesting with the skin lab that we are
Yes.
use. And we
Yes.
also have, I don't know if you're interested in the brain oxygenation directly, we have a field too.
Yes, I think there are many studies on this subject, but they are mostly done on water and on zebrafish so far. But for me, it's very interesting to go directly into the tissue. So yes, I definitely think that this would be interesting. For example, this is brain actually, and also this is water, and you can see here they measure the HO2, and there's a small difference, but this is statistically significant. The peroxide that is generated in the compound flash. So yes, but here it's water, and for water you have to go very high doses to see the differences. But this doesn't mean that in the tissue it will behave the same. So I think you need to experiment necessarily.
Thank you very much. It was a very interesting talk.
Thank you.
we learned a lot, so maybe we go to the second speaker.